Welcome back. So, today I am going to continue with the second lecture on proteins, and today I am going to discuss about protein folding in more details. So, we will see how proteins fold, we will see what are the driving forces that stabilize the structure of a protein. I will introduce the concept of folding funnel, and then I will briefly talk about diseases that are caused by protein misfolding, but this will be the major topic of the next lecture. So, in the previous lecture we have already seen this, that proteins, they have this particular hierarchy of structure. The sequence of amino acids in which they are connected is called the primary structure of the protein. Then consecutive amino acids can fold and form either a helical form or this extended form, which are called alpha helices or beta strands, and these are the two secondary structures of proteins. Now, in the three-dimensional space, these secondary structures can come together and form the tertiary structure of a protein. For most proteins, it stops here, but some proteins can also have this higher order organization, which is called the quaternary structure of a protein. The example that is shown here is myoglobin, where 4 polypeptide chains fold and come together to form this quaternary structure. Sorry, this is hemoglobin. So, what is a quaternary structure? Proteins containing more than one polypeptide chain exhibit a fourth level of structural organization, where each of these polypeptide chain is called a subunit. So, quaternary structure refers to the spatial arrangement of subunits and the nature of their interactions. Another example of a quaternary structure will be this. So, this is the crow protein from the bacteriophage lambda. So, it is a virus and it forms a dimer. So, the same peptide chain, it interacts with each other to form a dimer. So, it is again a quaternary structure. We have already seen hemoglobin. So, in case of hemoglobin, there are 2 polypeptide chains, the alpha and the beta. So, 2 alpha subunits and 2 beta subunits come together to form the tetramer called hemoglobin. This is a more complex structure. So, this is the coat of the rhinovirus, which comprises of 60 copies of each subunit. You can see that there are different subunits color coded differently, and there are 60 copies of each of these subunits, which forms this coat of the virus. In this case, it is a rhinovirus, which causes common cough and cold. We have already seen examples of proteins or large machineries with quaternary structures. For example, we have seen bacterial RNA polymerase, which has 2 alpha subunits, a beta and beta dash and an omega subunit. So, these 5 subunits come together to form the bacterial RNA polymerase. We also have seen bacterial ribosome, which has 2 big subunits, the 30S and the 50S, and individually these 30S and 50S are consisting of ribosomal RNA and 21 proteins. In this case, 2 ribosomal RNAs and 34 proteins. So, in this case, ribosome not only has proteins but also has ribosomes as its subunit. So, proteins are, we have already seen this, proteins are synthesized by ribosomes like this. In case of bacteria, the transcription and translation occur simultaneously, whereas they happen in 2 different compartments in case of eukaryotes. However, what ribosome does is it connects the consecutive amino acids depending on the mRNA sequence. So, when the protein is synthesized, it most probably looks something like this. But then how do we go from this to something like this? And this process is called folding. So, something like this, which is an unstructured polypeptide chain, how does it fold into a precise structure like this? So, this is the protein folding problem. So, in other words, we can think of it like this, that this is the extended polypeptide chain, and we have already seen this torsion angle. So, there is no rotation about the amide bond, but we can have rotations about the phi and psi torsion angles. So, if all these bonds rotate, then this extended polypeptide chain can collapse into a compact structure like this, a very precise structure like this. Now, one of the important question is that, is this a spontaneous process, meaning that the protein can do it by itself, or does it require help from other entities? We will see by the end of this lecture that the answer to this question is both are correct. So here is a simulation of the folding of a small protein. So you can see that it started with an extended structure, right? And here what we have done is we can see that the secondary structures are shown in magenta and yellow, the alpha helix and the beta strand. So you can see that the extended structure has somewhat collapsed and it is trying to form all these hydrogen bonds and tertiary contacts where these secondary structures are formed. So you can see that the 3 beta strands are forming here, and now it is more collapsed, and finally this is the final folded form. So if you run this simulation again, you will see that alpha helices are the ones which form first, and the beta, it takes more time to form the beta strand and even more time to form the tertiary contacts to get the final folded form. The reason for that is, in case of alpha helix, the interactions are very localized because the hydrogen bonds are formed between amino acids which are separated by only 4. So I plus 4, right? In beta strands it can be a bit longer, and the tertiary contacts can be even longer. So the probability of these interactions becomes lower. So alpha helices form first, then the beta strand, and then finally all the tertiary contacts. So now let's consider a very small problem. It's a thought problem where, let's say, we are thinking about a protein which is 100 residue long, and we know that for each amino acid it can have various phi psi torsion angle. So let's say in the Ramachandran plot, this phi psi torsion angle can occur either in the alpha helical region or in the beta stranded region or in the left handed helical region. So for each amino acid there can be 3 different phi psi torsion angle or 3 conformations. So if we consider a 100 residue protein, each amino acid will have 3 conformations. So for the first amino acid with 3 conformations, the second amino acid can also have 3 conformations. So together they can have 3 times 3, total 9 conformations. So if we keep doing that for a 100 residue protein, we can have 3 to the power 100. So 3 times 3 times 3, it goes on 100 times. So 3 to the power 100 different conformations, which equals to 5 times 10 to the power 47. It's a very big number. Okay. Now from experiments we know that these torsion angles, from going from one angle to another angle, it takes roughly 10 to the power minus 13 seconds. This is very, very fast. Okay. So let's say the protein, if it samples all these conformations and it takes this time to go from one conformation to another conformation, the total time taken will be this multiplied by this, which equals to 10 to the power 34 seconds or 10 to the power 27 years, which is actually longer than the age of the universe. Which means that if a protein which is only 100 residue long samples all possible conformations, it will never fold, right? And this is what is referred to as Leventhal's paradox, because this was first introduced by Cyrus Leventhal. So it means that, of course, proteins do not sample all possible conformations, like not even a small fraction of this. They sample very, very, very few conformations and go to the correctly folded form. Now, you might think that maybe I have taken up 100 residue is a big number, but that is not true. 100 residue is a very small number. There are proteins which have 1000 residues. Okay. So if we think about protein folding, we can divide all the issues and all the questions that are there into 4 major questions. The first question will be, how do proteins fold? That is, how do proteins achieve their final folded form? How do proteins know that this is the final structure? And how do proteins fold so fast? How do they reach that final folded form so fast? So most proteins fold within milliseconds. We have already seen the simulation. It happens within microsecond or millisecond. So it means that proteins are not sampling all the possible conformations. Then there is the third question, which is, can we predict protein structures without experimentally solving them? This is a very important question because as I mentioned earlier, that protein structures can be solved by 3 different experimental techniques: X-ray crystallography, NMR spectroscopy, and cryo-electron microscopy. However, all these techniques take a lot of time, lot of expertise, and they are also very expensive. So if we can theoretically model protein structures, then that will be very fast and it will be also less expensive. And finally, can we design artificial proteins to solve some of our problems? So there are so many issues that can be solved by designing artificial proteins. So I will tell you about the current status of where we stand in regards to these 4 problems. So let's take the first problem. To answer that, a very elegant experiment was done by Christian Anfinsen in 1960s and 70s, and that proved the thermodynamic hypothesis of protein folding. So what is the thermodynamic hypothesis of protein folding? It states that interaction between atoms in a protein control the folding of the protein molecule into a well-defined 3-dimensional structure. Or in other words, the primary sequence of the protein contains enough information that allows the protein to fold into its functional 3-dimensional form. So to show that, what Anfinsen did was, he and his colleagues, they used this particular enzyme called ribonuclease A. So I will talk about enzymes in the next week's lecture. And this enzyme catalyzes this particular reaction. So this is an RNA. It catalyzes the hydrolysis of this RNA into its subunits. A particular amino acid will become very important in this, which is called cysteine. So cysteine and cysteine, so it can occur in these 2 forms. So cysteine, its side chain has a thiol group or SH group. When this gets oxidized, it forms this disulfide bond. And when it is reduced, it goes back into this reduced form, which is the thiol form. So ribonuclease A has 8 cysteines which form 10 such disulfide bonds. So what Anfinsen did was, he took this ribonuclease A and he checked its activity, and he found that the protein has a certain activity. Then he used urea, which breaks all the hydrogen bonds and hydrophobic interactions. So we will talk about this in a few minutes. So it denatures the protein, it completely destroys the structure. And he also used a reducing agent like beta-mercaptoethanol to break all the disulfide bonds. So the protein becomes this. So it is not nicely folded like this, it becomes something like this. And then he divided it into 2 parts. In the first part, he removed urea first and then bubbled oxygen, and then he checked the activity. He found 100% activity. In the second part, he removed urea first so that the protein will collapse, and then he bubbled. So in the second part, he bubbled oxygen first so that all the disulfide bonds are formed, and then he removed urea. So in this case, he found only 1% activity. So he is doing the exact same thing. He is removing urea and he is passing oxygen. All he did was, he changed the order. So here he removed urea first and then oxidized. In this case, he oxidized first and then removed urea. Here he got 100% activity, here he got 1% activity, which was an amazing result, right? So why this happened? So let's go back to this. So when he removed urea first, the protein collapsed into this structure, which is the final folded form, and then he passed oxygen, which means he oxidized. So all the cysteines which are close to each other, they formed a disulfide bond and it locked the structure into this active form. So he got 100% activity. But when he didn't remove urea first but oxidized the protein first, the protein was in this denatured form. So cysteines, by randomly, they can form disulfide bonds. So this 1626 can form disulfide with 58 or 40 or 72 or any of this, right? So all possible combinations of these 4 disulfide bonds were formed, and only one out of all these possible combinations was correct, and only that one was able to fold properly when urea was removed. The remaining could not. It turns out that if we do our calculations correctly, there are 105 different forms in which these 4 disulfide bonds can be formed, and only one of those are correct. So 1 out of 105 gives you 1% activity. So how do we get this 105? So this is the calculation. We can form the first disulfide bond. So we are choosing 2 cysteines to form the first disulfide bond. So 8C2 will be the first. 6C2, now we are left with 6 cysteines. So we are choosing 2 out of those 6. 6C2, 4C2 and 2C2. So that's how we form the 4 disulfide bonds. Now it does not matter in which manner we form these 4 disulfide bonds. So which disulfide bond is formed first? So we have to divide it by factorial 4. And if you do your calculation, you will get 105. So 105 is the different ways in 4 different disulfide bonds are formed in the unstructured protein, right? And only one of those will be correct, which is again shown here. So only one of those will be correct. All these different disulfide bonds are formed. Only one is the correct form. So if I add oxygen first and then remove urea, I will get 1% activity. If I remove urea first and then add oxygen, I will get 100% activity. So what this experiment showed is that this protein can fold on its own. It does not need any help. So all the necessary information is present here. And this was actually shown by Anfinsen, and he got the Nobel Prize for this work in 1972. Since proteins can fold and since all the information are present in the primary sequence of the protein, can we predict protein structure if we know the sequence of a protein? And that is very important because it turns out that, so these are just some numbers, right? So proteins are made up of 20 amino acids. In bacteria, the average size of a protein is 300. So there are 300 amino acids per protein. Humans, we have 21,000 proteins. And if we consider all proteins on this earth, on this planet, there are 200 million proteins, right? Now, when we solve protein structures by experiments, it takes time, and in the last 70 years, we have solved only 2,25,000 protein structures, which is a very, very small amount out of all these possible protein structures. So it will be very helpful if we can predict structure from the primary sequence of a protein, right? So Anfinsen experiment has already shown that the information is there. Can we use this information? And this is what exactly many scientists have been doing for the last 20-30 years. So there is this particular competition, worldwide competition, that happens every 2 years since 1994. It is called Critical Assessment of Structure Prediction. So here all these groups worldwide who are working on this problem, they have developed their algorithm. So they are given sequences whose structures have been solved by different groups, but they have not been released. So they are given these sequences and they are asked to predict the structure, and then the predicted structures are compared with the experimental structure, and then some score is given. 90% score is what is the goal of this competition, that if you can get something that is close to 90%, it means that your predicted structure is as good as the experimental structure. So over years you can see that the best groups, so these are the best performance by any group, and it never crossed 40%. So that was the best that was achieved. In 2018, DeepMind entered this competition and they introduced something called AlphaFold. So it uses artificial intelligence, and you can see that it reached almost 60%. And then in 2020, the second version of AlphaFold came and they achieved almost 90% accuracy in prediction. So this is as good as experimentally determined structure, and this created a lot of interest. So it was predicted that this will change everything. So this AI AlphaFold 2 makes a gigantic leap in solving protein structures. And in the last 4 years, people have analyzed this. There are many more algorithms that have come up similar to this, and it turns out that AI can actually solve this problem very efficiently. So we can say that out of these 4 major questions, this third one is solved now. But then it does not answer the question how proteins fold, because what AlphaFold does is it takes the sequence and it gives you the final folded structure. It does not tell you how proteins fold, how it reaches that structure so fast, and what are the interactions by which it reaches there. So if we think about protein folding, we can think in terms of energetics. So these are the different conformations, and this is the free energy. So higher energy for unfolded structures will have higher energy, and the folded structure will have the lowest energy. So we can have all these different structures, and this is the final folded form. It turns out that the difference between unfolded and folded is not much. It is somewhere in between 5 to 25 kilocals per mole. In terms of time, we can think of it like this, that the unfolded structure looks like this, and over time it goes through all these different intermediate steps and then achieves this final folded form. So we have seen something similar in the simulation that we have seen a few slides back. So what stabilizes this final folded form? These are the few important interactions which stabilizes the folded form of a protein. There are hydrophobic interactions. So we have already seen that amino acids, there are hydrophobic amino acids like amino acids which have aliphatic side chain or aromatic side chain. So they form hydrophobic interactions, and it turns out that these hydrophobic side chains, they sort of go inside the core of the protein because they do not like to interact with water. Then there is hydrogen bond. We have already seen that in case of secondary structures. So alpha helices will have a pattern of hydrogen bonds, beta strands will have another pattern of hydrogen bonding interactions, and then there are electrostatic interactions between charged groups. So if this is a negatively charged side chain and if this is a positively charged side chain, they can interact with each other and they can stabilize this final folded form. So they can also interact with water and they can also interact with each other. So hydrophobic interactions, hydrogen bonding pattern and electrostatic interactions, together they stabilize the final folded form of the protein. What drives this final folded form? These interactions and also something called hydrophobic collapse. So it turns out that when in this unstructured form, these water molecules which are close to this protein, they have restricted motion. So they have restricted degrees of freedom, and also these hydrophobic side chains, they do not like to interact with water molecule. They like to interact with each other, which is also energetically favorable. So together, driving out of water molecules from here and interaction of these hydrophobic residues is something that is called hydrophobic collapse, and that drives the protein folding. So the protein will not sample all possible conformations. It will quickly go from this to this because of hydrophobic collapse, and we will have this final folded form. So this sampling of only few conformations is very elegantly shown by something called folding funnel. So we have seen this energy diagram. So free energy is plotted in the y-axis and conformations in the x-axis. So instead of just one axis, we can have two axes, x and y, for conformations, and the z-axis can be for the energy, and that will give us a diagram like this, which is called a folding funnel. So let's say the unfolded structure is here, which is energetically very high. Instead of sampling all these possible conformations, the protein will quickly go through only these few conformations and reach the native folded form, which is the lowest energy state. So it can start with all these different conformations and quickly go to this native folded form, right? So some proteins have a very smooth funnel like this, where the folding is very fast, and some funnels have this rugged funnel structure like this. So the protein will go through all sorts of intermediate states and finally reach the final folded form. So it turns out that if we think of bacteria, then this folding, this synthesis and folding can happen on the ribosome itself, because now we know that proteins can fold by themselves. So while the protein is getting synthesized, it will start folding, and then the complete folding is happened here. So this is the final folded form of the protein, and this is true for most small proteins. However, in some cases, if there are slightly bigger proteins or proteins with lot of hydrophobic residues, these proteins might not fold properly. So, or it can happen that most of the proteins, most molecules fold properly, but there are some molecules which do not fold. So in that case, extra help is needed, and that help is provided by other types of molecules. These are also proteins called chaperones. So chaperones, they bind to this unfolded protein and they prevent their aggregation, because when these proteins are unfolded, hydrophobic residues are exposed. So hydrophobic residues from one chain can stick to the hydrophobic residues of another chain, and they can form aggregation and they can precipitate. So they will not get a chance to fold. So that kind of aggregation is prevented by the chaperones. So they keep them separate and they give them enough time to fold properly and get into the folded state. Apart from that, there are another type of proteins which are called GROEL and GROES. So this is mRNA, this is ribosome, protein is getting synthesized. Now these chaperones, in this case we are seeing one particular type of chaperone called HSP70. So HSP stands for heat shock protein and 70 is its molecular weight. So it is 70 kilodalton in size. So this chaperone binds it and prevents it from aggregating. Now this chaperone will go out and it will allow this protein to fold and it will complete its folding. Some protein molecules might not fold properly and they will need additional help, which is provided by this large protein complex called GROEL. So GROEL looks like this huge barrel. It has 14 subunits. So you can see there are two rings up and down. So the upper ring has 7 subunits and the lower ring has 7 subunits. So total there are 14 subunits, and each of these subunit binds to this yellow thing which is an ATP. So ATP is hydrolyzed to ADP, which provides energy, and that energy is used to change the shape of this big barrel. So what it does, okay, I'll come to that. So this is a cryo-EM structure of GROEL, right? And you can see that it looks very similar to this, and then on top of that there is this small structure which looks like a lid. This is GROES. So GROEL and GROES help in protein folding like this. So this is the upper barrel and this is the lower barrel. So in the upper barrel, this is GROEL. It takes in the unstructured protein. This is misfolded or unfolded protein. It goes completely inside the barrel and then it will bind 7 molecules of ATP. Each subunit will bind 1 ATP and the GROES forms the lid. So the lid comes here. So this provides a complete separate environment for this protein molecule, and this is also referred to as an Anfinsen cage, right? So this prevents the interaction of this protein with other protein molecules. So hydrophobic interactions are prevented here. Now inside of this chamber is hydrophobic to begin with. Once this lid is closed, these 7 ATP molecules are hydrolyzed to ADP, and that energy is used to change the conformation of the 7 subunits, where initially the inside is hydrophilic and now it becomes, inside is hydrophobic and now it becomes hydrophilic. So this mimics the aqueous environment. Okay. Now since this is hydrophilic, it will induce this hydrophobic collapse because the hydrophobic residues would want to go out of this environment and they will stick to each other. So hydrophobic collapse happens and the protein will fold. So now we get this properly folded protein, and finally this folded protein is released from this chamber. So this is what happens in the top chamber, right? And you can see the same thing happens in the bottom chamber also. So this GROEL and GROES, it sort of acts like a double stroke engine. So in one stroke, the top chamber is folding one molecule. In the second stroke, the bottom chamber is folding another molecule, right? So what GROEL and GROES do is they provide an isolated environment for the protein to fold properly. The basic principle is exactly the same. The sequence of the protein determines its function, its final folded form. GROEL and GROES are not going to change the final folded form. They are not going to change the thermodynamics of folding. All they are doing is they are preventing aggregation and providing an isolated chamber where the protein can fold. So what we have seen so far is we know how proteins fold. We know how they fold fast. We can explain it by folding funnel. But again, I will not say these are complete. Still a lot of research is going on these two questions. The third one is more or less complete, And then comes the fourth important question. Since we know all of this, can we use this knowledge to design artificial proteins with unique functions to solve some of our problems? So let's think of it like this. The third problem was, if we know a protein sequence, can we predict its structure? So this is for the natural proteins which are found in nature. The fourth problem is sort of reverse of this. We have some function in mind. Based on that, let's say we have designed some three-dimensional structure of a protein. That's the machine. We think that this machine will do this function. Now, can we design a sequence which will fold into this three-dimensional structure? So we have some knowledge of this structure, or we have designed this structure. Now, can we predict the primary sequence which will fold into this three-dimensional structure, right? So this protein design or artificial protein, this problem can be divided into two parts. The first one is called protein engineering, where we take naturally occurring protein and we make changes into it so that instead of doing its natural function, it will do something else, something that we want it to do. So that is protein engineering. We are taking up an existing protein and we are engineering it to do a new function. And the second one is called de novo protein design. So de novo protein design means that this sequence does not exist. We are completely designing a new structure with new function. So I will talk about these in more details in lecture 20. So that will be the last lecture in the next week. So again, these are the books that I have followed. You can follow any one or more of these books. So that is all for now. Thank you.